Yeah, Anuj.
Hello, yes, sir. Hello. I hope so. I am audible to everyone.
Yeah, Anuj, you are audible.
Okay.
Did
So,
guest join or not?
steps also there. I am starting. So, good evening, everyone. I am Anuj Kumar Mehta on behalf of EcoFast Agri Solution. I warmly welcome to you all Day-8 of the Winter Nurture Training Program UNNAT KRISHI 2026. I truly inspiring to see so much energetic participation throughout this program. Your presence reflects your dedication to learning, innovation, and the advancement of sustainable agriculture. Today's session holds a special importance with agriculture rapidly transforming through genetic study and molecular techniques. It has become essential to understand how this advancement can be effectively integrated into sustainable farming practices. We look forward to gaining valuable insight into how genomic and molecular study of plant help in shaping the future of smart and sustainable agriculture. Today, it is a great honor to introduce our keynote speaker for today, Dr. Mohilan Mahendran. Dr. Mahendran is a notable academician with over 15 years of experience in plant breeding genetics and plant biotechnology. He currently serves as head of department and associate professor at Vivekanand Global University, Jaipur, Rajasthan, where he also lead the Agri-Business Management Department and contribute as a member of Academic Council, Board of Study, and University Research Board. He earned his PhD from CICY Merida, Mexico, and completed three prestigious postdoctoral research positions at UNAM, Mexico, one of the world top ranked university. His research spans molecular breeding in higher plants such as banana, turmeric, next generation sequencing, nanobiotechnology, antimicrobial study, and functional metagenomics. Dr. Mahendran has published 20 research papers, one patent, one book, and seven book chapters, and eight along with 89 nucleotide USA. He is a lifetime member of the International Promotion Scientific Community and has worked extensively with largest Musa germplasm bank in India and Mexico. Notably, he pioneered the development gene funded by IAEA Vienna. His leadership and innovation are reflected in 10 prestigious international awards at eight funded projects, including two major DBT projects in India, one SIDBI Solomon Chair Research Grant, and five projects from Mexico. He also guided numerous students and delivered six international radio interviews and acted as chief guest at several magistral conferences. We honored by his presence today and
Mr. Sivendra sir, CEO of EcoFast Agri Solutions, and he is our MOU partner also. So he is doing great things for the welfare of the society. So I am really honored to become the keynote speaker of this 21 days winter nurture training program, UNNAT KRISHI 2026. So I welcome all the participants. You can feel free to stop me at any time. Otherwise, you can ask me in the end of the session also, as per your wish. No problem. So today we are going to discuss about one small topic of my PhD research work I have done in Mexico. So can you please share the PowerPoint so all the participants can?
sure, sir. I'm sharing.
Yes, actually I have some small internet connectivity issue, so that is why I'm requesting. for the sharing my PowerPoint. So the title, as you can see, important markers. That means SNPs, single nucleotide polymorphisms, molecular markers for the starch metabolism in higher plants. In this case, we are talking about MUSA. So please go to the next one. So if you see the introduction, global population is already more than 800 crores. So currently, it is expanding day by day. So definitely in 2050, it will reach around 9.7 billion population all over the world. As you know, India is the #1 most populated country. After that, China and all other USA. We have to increase with the same or less cultivated area. This is the biggest challenge for us. So please go to next slide. So in this case, bananas and plantains collectively called as MUSA. Generally, people know only banana, but we used to call it as bananas, plantains, cooking bananas, and beer bananas. So totally, there are 4 types of bananas. Majorly, we call it as collectively we call it as general botanical name is Musa paradisiaca. But after that, I will explain what are the major and maize. Banana is the fourth most fruit only considered as a fruit. It is major food consumption in human consumptions. Especially if you go to Africa, in some of the major countries, they consume almost per kilo per day. So each person, each individual adult person can take at least near to 1 kilo. So imagine that much of importance of this crop. So all over the global production is more than 140 million metric tons they are producing, and it is one of the highest production plant, especially country like India. India is the #1 leader of the banana production. But what happened? We are consuming within the population, because as you know, Indian population is 143 crores. So we have so many bananas and we are consuming inside. We are exporting also, but when compared to the North American countries, the South American countries, we are exporting very less. Okay. So if you see the major food crops is very important additional income source also for the farmers, especially developing and underdeveloping countries. So as I told you, like humans, we have diploid chromosomes. So we can say that XX, XY. In banana, generally, the wild bananas are AA or AB. So they are the common ancestors. Apart from this, we are also having the T genome and S genome, but the evolutionary point of view, they are less important. So A genome and B genome, they are very, very important. So we have more than 1,000 accessions. If you go to supermarket, you may find maximum 10 different MUSA accessions, but we have more than 1,000 banana accessions. You can imagine. So I have tested all of them when I was working in ICR Institute in India, and it is one of the biggest germplasm bank we have in Tamil Nadu in South India. So the place where I have done my master degree, and I was working as project assistant also before I joined as a PhD scholar in Mexico. So if you see in this last line, you can see the plantains. So the cooking bananas and the East African cooking bananas and the desert bananas. So we have different kinds of banana varieties. Next, please. So you can consume the bananas in different manners. Either you can eat raw, otherwise you have to cook, otherwise you can bake, otherwise you can steam, and even you can ferment like beer bananas. I told you people are making beer also. So majorly starch is one of the most important biological components. If we see in the earth, it is the second most abundant biopolymer after cellulose. Cellulose is the wood wherever we are using. So starch is the second most biopolymer. In animals, including humans, we have the glycogen. So like in plants, they have starch. It is very, very important. So whenever the plant need energy, the starch will convert into sugar. And whenever the storage is starch, the sugar will convert into starch. So it is the continuous process. Most of the energy for the life comes from starch only. So if you see the starch in molecular level, you can see 2 major important materials are available. One is amylose, another one is the amylopectin. Amylose is the simple biopolymer. It is a linear biopolymer, but amylopectin is the branched one. It is like a tree, you imagine. So it is like a branched biopolymer. So next, please. So all of them comes under the family Musaceae. It is majorly 3 genera. One is the Musa, another one is Encente, another one is Musella. Most of the consuming bananas which we are having, all of them are comes under Musa. If you see in this table, you can see the NSET genus around 7 to 8 species, and they distributed in West Africa to Papua New Guinea, and especially we are using for vegetable purpose and fiber purposes. You can see the chromosome 9. Then if you see the Musa, genus Musa, majorly we have 4 important sexes. One is the Calimusa, Rhodoclamus, Australimusa, Eumusa. So Australimusa mainly we are using for the, you can find out in the Queensland, Australia, and the Philippines also you can find out. So you can see them as the root and the fiber also. Calimusa, Rhodoclamus we don't use much. Generally we use it in the ornamental. If you go to any 5-star hotels, any big parks, so there we can find out. So they are available in Indochina regions, Malaysia, Thailand, yeah. And also in Eumusa, there also we have some of the important species. So there you can find, including India and many places. This is the most important section I want to say. So it comes under the genome Musa and section Eumusa. There we find all kinds of things, either fiber or fruit or vegetable also. You can see the differences of the chromosomes. So minimum chromosome is NZA 9 and maximum is the 11 chromosomes, Rhodoclamus and Eumusa. And the Calimusa, Australimusa, they have the 10 chromosomes. If you see the humans, we have the 23 pairs of chromosomes. I know, I think most of them will understand. Okay, next please. Previous. Actually, one more third genera is also available, Musella. This is a controversial one because the insect Sedis is a new section ordered in the genus Musa. From our previously, she was the director of National Research Center for Banana, NRCB ICR Institute from Dr. Uma. She was the director also. I know her personally, but she got retired now. So she is the one person who bring to the notice to many. Then in 1947, the section we have already seen this. So there are more than 1,000 cultivars available in the worldwide. So this is one of the important fruit crops for the many countries. But we don't know the cultivars. Cultivars means we are consuming directly. We are also having the wild bananas in the nature, which means we have find out the seeds of the bananas also. The banana size are very less, and some of them are acidic conditions. It is not suitable. People don't like. Majority of the people don't like. But whenever we are going for research purpose, whenever we are going for the jump blossom, we used to taste it just for the curiosity purposes. Next, please.
Yes.
Yes. So here you can see the wild bananas, how they are naturally they are arranged. And if you see the right side picture, you can see the Musa velutina. This is the wild species, but you cannot consume so much because the taste is not that much good. But people are using it for the ornamental purposes. So you can see the wild species, they are producing seeds, fertile seeds. You can go and you can plant it or broadcast in your land. Definitely it will grow if they find out the suitable conditions. So there are so many plants they are available, but only few number of species they are domesticated, and they are interrelated to the wild species also. Why we are maintaining jump blossom? Because whenever there is a disease outbreak comes, definitely we can go for the wild bananas, and definitely in future many researchers they can also go and study. They can create new cultivars. So according to the population, we are call it as population genetics. So Indian population they love some variety of bananas. For example, Rastali, Puvan, Nei Puvan. These are some of the names scientifically we call it as. So, but if you go to Mexico, for example, so there people are saying Manzano, Platano Macho. So these are some of the local names. They call it as for some of the varieties. Next, please. If you see the evolution of the cultivated bananas, as I have told you, so they Musa acuminata and Musa bulbiciana, they are very, very important. If you see most of the cultivable bananas, they are sterile. They are producing fruits without seeds. So there is parthenocarpy. And in banana, generally we call it as 3 different ploidies. One is diploid, like humans, 2 parents. Another one is triploidy, 3 parents. And the tetraploidy, 4 parents. 45 kilos or 50 kilos, very strong plants they are producing. especially FIA, which comes under Honduras. Honduras is a small sized country which is available in South America. Though there, the scientists, they produce so much of FIA bananas. So mainly they focused for the industrial purposes. If you taste the banana, you don't feel the same kind of taste what we are tasting in generally. It looks like a potato. Okay, you don't feel anything. You don't feel any taste of banana. But generally, we use it in the world for the industrial purposes. So you can see all kinds of banana. Whatever bananas we are consuming, they are diploid and tetraploids. Okay, so there in the right side below picture, you can find out these are some of the important wild species which are available in the world. Till now, also you can find out in many parts of the world, and we are also having, especially they are these wild species. They originated from Southeast Asia, including India. Next, please. Yes, here in this picture you can find out easily the red colored ones. They are all Misa acuminata, and the blue colored ones, they are the Balbisiana. So these are the major contributors of the today's bananas. Next, please. So FAO, Food and Agriculture Organization, they are very, very important for the world. So they are producing, banana is produced in more than 135 countries. Okay, this is very, very important. Tropical and subtropical regions. As I have told you already, India is the #1 highest producer in the world after the China, Philippines, Colombia, Indonesia. These are the countries. But we are consuming most of the bananas within the country. So if we especially see in India, we are producing 30.81 million metric tons. It is a huge for the Indian population. It is also a very important population. So this data we took from Food and Agriculture Organization statistics 2022, 4 years back. Next, please. Yes, this is very complex pathway. So starch and sugar metabolic pathways. As I told you, this is the anabolism and catabolism. So there are more than 20 genes which are involving in banana. Next, please. Yes. So if you see the biosynthesis of starch, there are more than 20 genes, but these are the major 7 key genes which are involving. So AGPase, sucrose synthase, starch synthase, starch branching enzyme, debranching enzyme, polyolase. So these are the important steps, key major genes which are involving in the starch metabolism. Next, please. Yes, so chloroplast. So chloroplast is very important for the photosynthesis of bananas. So whenever the starch is produced, there are 2 kinds of starch. One is the transistor starch, another one is storage starch. So in chloroplast, you can find out the transistor starch. This particular starch, which is useful for the plants whenever they are doing the photosynthesis, the respiration, all kinds of physiological activities. If you see the storage starch, which is available in the leucoplast and the amyloplast. So whenever the plants generally they store their starch material, storage starch in their fruits or wherever it is available based on the plants. For example, if you see the cassava, they maintain in their root systems. So based on the plants, they are maintaining. So if you see this picture there, you can find out the amylose and amylopectin structures. Next, please. Sorry, glucose. That is the glucose structure. Next, please. Yes, so leucoplast, so this is just general information I gave. It is not that much important. Next, please. Yes, plant breeding also, this is also very important nowadays. Previously, farmers are using the conventional breeding, which means the farmer can go and by naked eye, by visually they observe and they find out which is the best plant to use it. And then nowadays we are going for the breeding programs. So in this system, you can find out how the plant breeding is come to this very, very important system. Nowadays, we are using the modern tools. We are not using much of the biochemical markers. We are using the molecular markers, which are very accurate, 100% precise. You can go and you can save your time and you can get the results immediately. Okay, without any second thoughts. So there are so many technologies we are using nowadays. This is one of the technique that we can see further. Next, please. Yes. Why we have to use the molecular markers? Because we can easily find out the genetic loci, which is generally control the stress tolerance, and no need to wait for the phenotypical. Because otherwise, what will happen? Banana, it will take minimum 1 year. Some of the varieties it will go up to 1.5 years also. So no need to waste your time and space, and no need to wait for the field testing. Whatever we do, we can do it in the lab within 10 to 15 days maximum. So, and it is more precious also for the gene pyramiding and integration. And transgenic approaches also we can use, which means we can control the gene regulations, up regulation, down regulation we can do. And also we can use it for the gene transfer. And most important that I want to say, it is the polymorphic, which means same gene which can be available in different places, like stem, root, leaf, different kinds of fruits, wherever we can find out. So and also we can find out for the traits of interest, which means in this case starch metabolism. Next, please. Yes, so single nucleotide polymorphisms in the genome sequences of the individuals, we can find out in the whole population or maybe in the species. In a particular nucleotide, in the coding regions, a single nucleotide can be changed. So that's why we are saying SNPs. So it can be used for so many aspects, especially in plant breedings we are using nowadays. Okay, there are so many other aspects also people are using. Next, please. Yes, so a couple of decades back, people are using the other traditional markers like RFLP, REPD, AFLP, okay, because they are available in the abundantly, they are available and they are cheap, and they also showed high polymorphism. But nowadays, SNPs also create alleles and haplotypes, so they are more precise than the previous traditional molecular markers. So we are using with the help of only one particular nucleotide. It may be indels, we used to say, either insertion or deletion of a particular open reading frame, or it may be a substitution, or it may be inversion. But also we want to.
ये एस्ट्रोस होगा इसको बोलेगा
पहले किसका था शेड्यूल में किसका था
पता नहीं किसका था दूसरे का था
दिलीप का था
पता नहीं किसका था दूसरे का था
तो तो बोलेगा
एस्ट्रोस बोलेगा
हाँ तो बोलेगा एक बार
Okay. So SNPs, very important aspect is either it can change or it cannot change the protein structure also. So this is also very important. We cannot say that one particular place SNP is available, maybe insertion or deletion or maybe inversion. We cannot say that is very important. Maybe it may change or may not change the protein. If the protein change is possible and if it is very useful, then only we can consider that is the important SNPs. Okay, so why we are saying this? Because whenever we are seeing different kinds of mutation, it happen due to somatic mutations and evolutionary process. So genetic diversity is also very high, especially in the cultivable bananas and wild ancestors. And also they have the genetic variations, and they are very useful for the plant breeding and other recent technologies. So that's why we are using, in this case especially, we are going to check it out for the starch related traits, starch metabolism traits. We are going to find out in both bananas and plantains. Plantains means cooking bananas. Bananas, I want to say that we are consuming directly, like gross missile or cavities. Next, please. So the principal objective of this work is to develop a PCR based SNPs markers. No, next, please. Next. Yes. So the principal objective of this work is to identify, develop the PCR-based SNPs markers for the key allele genes involved in the starch metabolism in MUSA general types, and the alleles should be associated with the known traits. In this case, starch markers. Next, please. So what we are doing, we are going to select the key metabolic genes which are representing the starch metabolism, and the MUSA accessions also we are going to select the representatives, because we don't have the time to do for all the thousands of accessions. So we are taking the 10 important MUSA accessions, and we are going to check the starch characteristics, and also we are going to extract the DNA. Then we are going to use the primers also, either degenerative primers or specific primers. Then blast searches in bioinformatics analysis, we can find out the databases already what is available. in the NCBA, National Center for Biological Information, EMBL, European Molecular Biological Laboratory, then DDBJ, DNA Data Bank of Japan. So to find out the homologous sequences, then we are going to design the premise from DNA and mRNA, messenger RNA. Then we will amplify, clone the sequences and assemble, edit and identify by using the BLAST. And also with the help of using Mega software, we can going to construct the phylogenetic trees. And then after that, we are going to find out what are the SNPs associated with the phenotypic traits. And putative, they should be useful as a putative SNPs markers. Then we are going to useful for the starch traits. Next, please. So what we have done, we collected the DNA, deoxyribonucleic acid. So from Musa Gemplasmang, which is available in the Usmal, Yucatan, my PhD guide who invented that one. That was one of the biggest Gemplasmang in Mexico. Then after that, putative markers we have seen already. We are going to search in BLAST whatever available in the databases. Then primers will be based. Then next, please. Then once the primer arrive to the laboratory, we are going to check the proper temperature and concentration, all kinds of things with the help of gradient PCR. Then after that, once we finalize it, we can go and multiply in normal PCR, polymerase chain reaction. Then after that, we identified the PCR products, especially 0.5 to 1.5 kilobase per, which means 500 base pair to 1500 base pair. Because that time this project was started in 2010 to 2014. So imagine that time it was very difficult to sequence the DNAs because they will denature, because that was the tropical area. And in Mexico, we don't have that much sufficient. So we used to send the University of Arizona, USA. So then after the PCR products, we cloned and we transformed, and then we sent to sequences. Then next, please. Then after that, phylogenetic analysis with the help of Mega software. So neighbor joining method were used. So NG algorithm was implemented, and then we have the using the Poisson correction also. Then bootstrapping also used in order to grouping a particular pattern in the phylogenetic tree. Then after that, SNPs, we found out the indels and SNPs to develop the PCR based markers. Next, please. Okay, this one also the same thing what I explained already. Next, please. Okay, so the this is very important thing. So we received the most of the plants from ITC, International Transit Center, which is available in the Catholic Leuven University in Belgium. So this is how, in the right side picture you can see, this is how you can receive. All over the world they are sending us free of cost. Only you have to bear the sending cost. That only you have to charge, but they are sending free of cost all the plants all over the world. This is also one of the important germplasm of Musa. And also we have in Tamil Nadu, that comes under ICR Institute. Then we have in Belgium. So ITC, we received most of the plants. Why they are sending 5 tissue culture plants? Because sometimes by human error or somoclonal variations, sometimes 1 or 2 plants may or may not have some mutations. So in that case only they are sending you 5 replications, and whenever you are going to transplant them in the field, you don't know what is the field conditions. Maybe some places so hot, some places very cold. So you don't know how much will survive. So that is the major reason they are sending you 5 replications. Next, please. Yes, in this picture you can see. So in the left side, I took one plant. It is my hand. There you can see this is the wild banana with the seed. So I just broadcast in our field during my PhD. So that time that wild plant, one of the wild plant grown in my hand, that you can see. So right side you can see the DNA isolation from the Musa accessions with 1% agarose gel. The major challenges that we can find out in the Musa, they have so much polyphenols and polysaccharides, so they will not allow you to extract the DNA. But we need the proper DNA, which means the high quality and high concentrated DNA also. So then only we can go for the sequencing purposes. Otherwise, when you get the sequences after the sequences, you will in the chronogram you will not get the proper sequences. So definitely this will affect your analysis. So there is a major challenges. Next, please. Okay, these are the 10 MUSA accessions we used for the study. You can see in the picture 9 MUSA accessions comes under the UMUSA. You remember that previous table that I mentioned. Most of them are comes under the UMUSA. Then Australian MUSA also we utilized for the ornamental purposes, also for how it will go in the MUSA Beccari. Okay, so some of them are wild banana, some of them are cultivars. You can see the genome also. Some of them are AA genome. Some of them are AB. Some of them BB. Some of them are AAA. Some of them ABB. You can see. So and they have the ITC numbers also for most of them. Okay, only one we collected from the Mexico. Next, please. Yes, in this picture you can see how we retrieved the sequences. Actually, it is the AGPS, large subunit gene fragment. From the consensus, we have designed the forward primers and the reverse primers. So only one particular sequence which is available in the MUSA germplasm bank. That star I mentioned in the number 11, you can see. So that was also we have taken, and we compared with the other dicards and the monocards also. Below I have put it all the sequences with their NCBI gene sequence number with scientific name also. Next, please. Yes, this picture is very important because from here 10 accession 10 clones we have made for the each plant, because we don't know what is the gene family. Actually AGPS is a big gene family, but still we are still doubt on how many polymorphisms and how many isoforms which may be available in each and every accessions. That is very important. That's why we took 10 for each one. So 10 accessions means 10 into 10, 100, but some of them are very, very less and contaminated. That's why we eliminated. So that's why we come to the conclusion as 89 sequences. We can go further analysis. So in this picture, you can see number 3 variant. You can see the similarities between the plants. Okay, so this is the first 3 exons and 2 introns. So here you see the variant 3. Variant 3 is very, very important sequences. So 90% of the all 3 exons they have shown. So I give the number of nucleotides also, you can find out. And then number 1 and 3 is 90, 70 and 80. Number 4 is the least represented variant. So out of 89, we come to the conclusion of 15 haplotypes and 4 variants. In the further slides, I will show you. Next, please. Yes, so this table is also very important. You can see the number 3 variant. So out of 89, 46 clones which are comes under variant 3. That's why which is very, very important isoform. And then variant 2 consecutively you can find out 19, and variant 1 comes under 15, and variant 4 is the least observable variant which is available in the all the sequences. Next, please. Yes, this is another analysis, dot matrix analysis. Here you can verify that variant 3 we can find out the full similarity. So almost 90% similarity. This is the 100% identical when compared to the original systems. So the number 1 and 2 they have some small differentiations, and number 4 is the very least available of the indels and other changes. So you can find out in the inversions of the perpendicular lines. Next, please. So next, by using the Mega software, we can find out the this one dendrogram we created. So here only we have all the 89 sequences. We did not include any other outside plants. So here you can find out the second exon, which is very, very important consensus. So there we find out the major clade which went to clade C, which means the variant 3. And after that variant 2 and variant 1 and variant 4 finally. Next, please. Yes, this phylogenetic analysis, here we included all the externals, which includes the monocards and the dicards. So here you can see we included purposefully for some of the poils. Poils means wheat, rice, rice, wheat, maize, all the grass family. Because they are also very close to gingiberales. So that's why we include that. So if you see, they are also coming under separately. And then all the variant 2 and 3, they comes under the different clade. And also outgroups, they went separately. So from this, we can find out the variants comes into 4. Next, please. Yes, so when this project was designed, there was no release of the banana genomes, but after 2012, A genome was released, and 2013, 2012 A genome released, 2013 B genome released. So that's why we also utilized how much we can compare with our genomes. Next, please. Yes, so in this very important, because Genoscope with the help of CIRAD, that is a French people, they analyzed so many years with the help of very big international team. So here, this is the A genome. You can find out the 523 million base pairs of A genome. You can find out 11 chromosomes, and the total number of genes is 26,829. B genome is little bit less. So now it is available for all over the world. Anybody can go and they can check, they can analyze with their gene sequences. Next, please. Yes, so you can see the 11 chromosomes for out of 11 chromosomes, we can see 3 chromosomes of our 4 variants, especially variant 1 and 4. You remember the dendrogram and phylogenetic analysis, they all comes under the same clade. So here you can find out in chromosome number 4, and variant 2 comes under chromosome 6, and variant 3 comes under chromosome 9. So you can find out large subunits, all of them they available in the chromosomes. Next, please. Yes, here you can see the what type of variant, which peptide or chromosomes, how much percentage which is available. You can see variant 1 and 2, they have the 100% and 96 and 98%, and variant 3 and 4, they have the 100% similarity. Okay, I think everybody understand the difference between identity and similarity. Identity means the same nucleotide which will be available. Similarity means it may be second and third triplet codon may be different, but same amino acid will be present. Okay, next, please. Okay, after that, this is very important. We analyzed all the sequences with the help of sequence software. We uploaded all the sequences, which I told you, 89 clones, and come closer into 15 haplotypes. From this, further divided into 4 variants. So all the 89 sequences which were uploaded in NCBI, you can check and you can retrieve. You can analyze also with your sequences. Next, please. Yes, this is, we come to the conclusion. So SNPs we have found out in the AGPS large subunit in Musa. So when the starch phenotypes associated with the variants, SNPs based molecular markers can be designed very easily and very fast. And especially this will help for the MAMA PCR, which means mismatch amplification mutation assay. So this is first developed by Shiverstone and Braden. So this is MAMA PCR is very, very important whenever you want to go to the field and you can take the samples and you can analyze within 10 to 15 days. And another one also, technology also that time was very famous, is tilling, which was created by Dr. Till. So that is also another technology we can use. So this strategy, we can usually find out how many number of variants which is present, and how many of them are actually going to translate into proteins, because some of them may be the pseudo variants. So that also we can easily identify. So this is very, very important for the basic research, science research, and also banana breeders and biotechnologists whenever they want to manipulate the starch traits, which means in this case they want to say about the amylose and amylopectin. If you down-regulate or up-regulate a particular gene, particular enzyme, definitely the taste will change. So people may like it in future. So SNP-based molecular markers can be linked to useful traits for accelerating crop improvement. So this is banana generally used as orphan crop, which means the crop which is consumed more, but we don't have sufficient data. So that banana also we can use, and other modern crops also we can use. Next, please. So this project was funded by the IAEA, International Atomic Energy Agency. So we are having the international genome research. So we are the first people who analyzed in this AGPS of Musa, and we published in 2014 in Elsevier journal. Next, please. Okay, last but not the least, I would like to thank to my PhD guide, Dr. Andrew James. So I was his last PhD candidate. After 2 years, he also got retired. And my co-guide, Dr. Jorge Humberto Ramirez, he's the bioinformatics scientist. He also helped so much. And then engineer Ivan De Hone, he's the curator of the ITC, Musa International Transit Center in Leuven, Belgium. He also helped us so much whenever he's sending me grateful to the plants, all the tissue culture plants. Then INIFOB is very important scientific research like our ICR. So they also helping us. They gave the land free of cost for our research, for our installing our own germplasm bank for our research institute. So and two technicians, they helped each and every time. Mr. Jose Roberto and Ms. Jeremy Minero, they helped. And this is my research institute, CC, Centro de Investigación Científica de Yucatán. This is the Central Government Research Institute, Government Public Institute. Then CONACYT, that is the funding agency for my PhD. Throughout my PhD, they gave the money. And PROMUSA, I'm also one of the member of International PROMUSA Scientific Research Community. And also IE, International Atomic Energy Agency. So nowadays they are using atoms for peace. So that's why they are also funding so many research projects in global level. Next, please. So these are some of the my team members and all my friends, those who helped me very much. So fortunately, I went to Cuba also during my PhD. So I stayed there for 1 week to attend the international conferences, and so many conferences we attended nationally, internationally, and we did really good work. And I'm really thankful to all of my friends and all of my research community, those who helped me. These are small thoughts. Thank you very much. If any doubts, you please feel free to ask me. I will try to answer yours.
Yeah, thank you very much, sir, for such an enlightening, forward-thinking session. Your insight in agriculture, biotechnology, and sustainable farming have truly expanded our understanding for the future of agriculture. Now we would like to open the floor for interaction. Participants are encouraged to ask their question and directly engage with Dr. Mahendran. Dear participants, you can unmute your mic and ask your questions, please. If any doubt, you can ask directly to the sir.
Any doubts?
Are you listening?
Any doubts?
Are you listening, sir? One question arises in the chat
Yes,
box.
please. Yes, yes. Can you please read? Because if I going to read now, I will go out of the meeting.
Okay,
Can you
okay.
please?
I am ready, sir.
Yes, please.
One question is, sir, please explain MAMA PCR.
Acha, okay. Actually, it is the MAMA PCR, mismatch amplification PCR, mismatch amplification mutation assay, which means I will tell you very easily. So, for example, we have 536 nucleotide number. We have the adenine. Okay. So, if you know adenine is there, definitely the protein is going to increase, for example. If there is no adenine, if there is cytosine, for example, okay, adenine, guanine, cytosine, thymine. So if there is no adenine, if there is cytosine, definitely you will not get the amplification in PCR whenever we are doing the PCR. Na? So because this is MAMA PCR, mismatch amplification mutation assay, which means, for example, let's say you don't have the variant 3. We can say from our research work, variant 3 is the most important variant, which is very, very important for the plant. If there is no variant 3, plant will definitely die. But let's say variant 4 is not that much important. So if it is available in there, so definitely your protein will not express and it is not important. But if you have the variant 3, definitely you will get the amplification and you can clone, you can transform, you can easily transfer into another plants also. So that is why Mama PCR is very, very important. So you don't need to waste your time. For example, you know variant 3 will work definitely. So you can check your PCR. So PCR maximum it will take 2 hours. Okay, so then you can run in the gel, then you can do the cloning, you can do the transformation. Less than 1 week, definitely you will get the 100% accurate result. Then you can go for the field. So that's why this kind of molecular research is very important.
हेलो सर।
Yes.
सर मैं हिंदी में पूछ सकती हूँ?
Okay, okay.
सर आपने बायोइन्फॉर्मेटिक्स का ये इसमें इंक्लूड किया था, टूल वगैरह जो दिखाया उसमें सीक्वेंस। तो मैं पूछ रही हूँ सर, इसको मशरूम में हम कैसे अप्लाई कर सकते हैं, लाइक उसके द क्वालिटी इन्हांसमेंट के लिए कौन सा सॉफ्टवेयर या मतलब जो टूल होता है ना बायोइन्फॉर्मेटिक टूल,
Yes.
कौन सा यूज करें सर उसके?
Actually, you have to go to the NCBI. So there you can find out the nucleotide or genes or proteins. Please go there and study which type of gene is very important for the quality enhancement or taste enhancement. There only you can go and you can study. Okay. So there are so many softwares is available in bioinformatics. It is a very big field also. So there are so many softwares available. So based on what you want to improve, we can do that. If you want, you can contact me also. So we can also think and we can come to the conclusion. Okay.
How can modified their sequence?
Can you please repeat? How can modify?
How can modified their sequence?
No. I'm not saying that we are going to modify the sequence. I am saying first we are going to analyze the sequences. Either it is nucleated sequences or protein sequences. So there, after that only we can come to the conclusion. First we will get the basic informations. Then only we can go and modify.
Thank you so much, sir.
Hello, sir.
Yes.
Sir. Good evening, sir. I am Vikram Singh.
Okay.
Sir, such technology also used in only fruits crops or others food grains crops also.
Everywhere we can use, no issue, because you know all kinds of living organisms we are having more or less same nucleotide sequences. If you see one interesting fact that humans and bananas, they are sharing 50%
Same.
of
DNA
the
of the same.
gene sequences. Yes.
Yes,
Any
sir.
plants we can use, no issue.
Okay, sir. Thank you, sir.
Thank you.
Any questions? I hope so. All are clear, sir. Okay. Thank you, sir, for patiently addressing all the question and providing such a practical.
Anuj, kindly see the message box. There is question.
Okay, sir. Yes, one question. Are there variety released for tea to grow well in plains?
For tea, yes. In tea also, actually, I told you now in DBT, we received one multi-institutional project, which is 3.6 crores. So there also people are using so many molecular markers, especially SNPs markers they are using. So there are so many research groups, they are working in India, especially Tamil Nadu Agricultural University and Assam, and so many research organizations, they are working actually multi-institutional projects. So especially nowadays they are using SNPs markers for the abiotic stress also, because you see hilly regions they are growing, because they are also facing so many other issues, abiotic stress and also biotic stress. So it is so much challenging for the tea, especially India also we are producing so much tea because of the Britishers. So they converted all the major forests into tea plantations. So they are also doing, especially turmeric also they are doing, tea also they are doing. Most of the cash crops and cultivable crops, horticulture crops, they are using SNPs. And they are releasing varieties also in India.
Okay, sir. Thanks.
Thank you.
Any
Hello,
other
sir.
question?
Hello. Hello, sir.
Yes, please.
Sir, you told, sir, that is the triploid varieties of banana are sterile in nature, but the consumable banana are diploid and tetraploid. But the we go for propagation by only rhizomes, sir. But as per diploid and tetraploid, actually they will seed set, na, sir.
No, actually, wild bananas only have the seeds. But nowadays we are having thousands of accessions, na. So what happened? Farmers they make them, and nature also make them. Nature also make them.
Okay, okay.
Yes, yes, yes. But we can also go. That is the purpose of germplasm bank. Any germplasm bank, not only in banana, in any germplasm bank we can go. For example, I forgot the name. There was one particular fungus which was devastating fungus. They destroyed one particular banana variety in 17th and 18th century. Most of the world population they utilized this particular banana. But what happened? One particular fungus they destroyed. I think Fusarium, if I am not wrong. Fusarium. I think it destroyed the whole banana variety. Now we don't have it. We lost. So in those purposes only we are maintaining germplasm bank. So if we know, we can go and we can create new varieties. No issue. It is open to all. Open to all. We can go. Only we need the prior permission from the particular research institute director or whatever. Then we can go for the research purpose. Definitely they will allow. We can do individually as a company, as a research organization or school, college or university. Anybody is open. It is open for everybody.
Any other question, participants? You can write your question in chat box also.
Sir, one more question.
Yes, please.
So while going traditional method for observation of whether the variety or tolerance for stress or drought, it will take a long time. In case of molecular, you told that, sir, it will take very less time.
Yes.
And how